Very good. Hello, everyone. This is Emil Kakkis with Ultragenyx, and I appreciate the introduction and opportunity to talk to you a little about my journey translating rare disease research into medicine over the last 30 years. I'll try to give you a sense for what it's like to start at the beginning, take science and turn it into something that's useful for people, and to understand how academic scientists and the industry have to work together to get drugs developed, that no one by themselves can actually make this happen the way it needs to be. And so I hope to give you a little bit of also insight into the practical natures of this. And finally, I want to talk about what are some of the challenges in getting rare disease drugs, including what might be challenges, particularly for variable heterogeneous neuromuscular disorders, and which might, of course, apply to the mitochondrial field, where getting treatments developed can be very challenging. So thank you for the opportunity to speak to you today about my history in translating rare disease research. Early in my career, I had the opportunity, beginning in 1991, which is where the 30 years began, to work on a project with Elizabeth Neufeld to develop a treatment for MPS I. The story is complicated, but this is a picture after the first 6 months of the treatment trial, and with 9 of the 10 patients in the trial, a heterogeneous group of kids that all had the same defect, essentially a deficiency of an enzyme, but they could be as severe as a child in my lap or as mild as a woman standing behind me. And the difference between those two was maybe a few fractions of a percent of normal of the enzyme. The challenge in developing a treatment for a disease like this is how hard it is. But what's been great in these last years is how many new technologies and new approaches for treatment, ever more sophisticated and involving the whole cell biology of our bodies, has put us in position to be able to treat many diseases, and there's been extraordinary progress. But it has also been quite the challenge. Now, many people trace the beginning of this move toward developing treatments for rare diseases to the Orphan Drug Act in 1982. The Orphan Drug Act was US legislation that was provided some exclusivity and tax breaks for investment. But if you really look at the first 10 years of the Orphan Drug Act, not a lot happened. There were a few small molecule drugs that were repurposed, but there really wasn't much in what I would call complex, sophisticated, disease-specific drugs being developed until PEG-ADA and Ceridase for Gaucher. Those were the first. The truth is, making those kind of drugs was extraordinarily expensive, and there'd be no way to be able to fund that development with such small patient populations. And the truth is that the cost of these drugs were what helped drive the potential to get there. Now, to do other treatments, there was much more technology was needed than what was available at those days, which was basically enzyme replacement therapy was the one approach that was working. So if you look over the history of time now, from that early period, the first 10 years were mostly, as you can see, small molecule repurposing. There are very few biologics. Most of the biologics were blood products that were, purified rather than recombinant products. And then there's this surge that starts to happen around year 2000. I'd gotten through MPS I enzyme therapy by 98, 2000, but the drug wasn't approved till 2003. Then things started to take off. And the question is, why is this happening now? And people thought it was all about the Orphan Drug Act, but honestly, there's not that much happening the first 10 years. There's so much more lately. And there's a number of factors, I think, that are pushing us forward. I've talked about advanced technology. Yes, that is helping dramatically. There's been some other drug legislation and regulatory policy and guidances published. And there have been some improvements in understanding how to do clinical development. But the thing that keeps it moving is the fact that there's been a significant financial potential, even in ultra rare disease. The treatments are expensive, but the total value then is sufficient to be able to fund 100, 200 million plus of clinical development costs to actually get a drug approved. If it wasn't for that, it wouldn't be happening. And the thing that happened in the 1990s is that Ceridase, in fact, was doing well. It was changing lives. It was very expensive and attacked for that, but the truth was it helped create a business model for how you might be able to afford to develop treatments for rare disease drugs. So the financial potential becomes an important part of what's happening. And you need to understand that to understand why, if you don't have it, how hard it will be to go back to where we were before, where you were not being able to invest in the kind of money required to get a drug developed. Now, my own experiences involving academic partnerships are shown here. Of course, the first project, Ultragenyx, was me working with Liz Neufeld, and I bridged both the academic world and the industry world. Then after joining BioMarin, I did a number of projects with different academic investigators. I'll touch on some of these. And then later, starting a new company, Ultragenyx, we did a few more. But if you notice through all these stories, there's always an academic innovator who's thinking up a way. But to make it real for people, you have to go all the way to the end and get through the development, which takes so much more money and so much more time to actually make the drug an approvable drug that someone can actually take. Now, this is, the history of my life in the first few years. I started as an academic professor, or as a fellow, actually, in 1991. That was my bungalow. I always talk about this bungalow because just to talk about how things can be in the beginning, these were cheap lab spaces that were available to us, and they weren't well maintained, but you could do work there, and I had plenty of lab space. I just didn't, it wasn't exactly, the finest lab facilities, but they were workable and we got things done. Developed a manufacturing process by 1992, created a cell line, and kind of paced together a way to make milligram quantities of enzyme. I then treated dogs and were able to show that this really worked. And the dog B below is the one treated, stand straight, ran around, was healthy. And we showed that small amounts of enzyme worked. Then we got stuck. A patient group, Ryan Foundation, for their son, Ryan Dant, started raising money, helping support our work. And we were able to get to the clinic once BioMarin joined the effort, then affiliated with Genzyme afterward in a deal. And in the end, it took, on that picture with all those people, those are the people at BioMarin that had to touch the product in order to get it approved. More than 100 people had to do various things for the product to get it approved. And the amount of money and the number of people was staggering. In the early days, it was me and I had a few people, but the truth is, to get to the end was dramatically more money, well more than 100 million spent, which I never thought was possible for a treatment that has less than 1,000 people in the world. That's sort of the first story, and my first opportunity to be both on the academic side and then on the company side. Now, following that opportunity, while at BioMarin, I took the opportunity to meet other investigators like myself who had been doing work for a while and couldn't get anyone to work on their rare disease program. And there were a lot of them. One of them was John Hopwood here, a giant in the field of lysosomal diseases, particularly MPS, had cloned a number of the enzymes. And he had an MPS 6 enzyme therapy, which worked well in the cat model. And two companies started to stop that program, but the enzyme was incredibly difficult to make. Incredibly difficult. It was 2 or 3 mg per liter. What you have to understand, when you're making like an antibody in recombinant culture, it's 1 or 2 grams a liter, right? When you're making an enzyme at only a few milligrams a liter, the cost of making the enzyme is extraordinarily high. And for MPS 6, it was extremely high. 2 companies started to stop because it just wasn't economically viable. And we had to develop a very difficult perfusion culture system to tune up and get enough enzyme made. Now that's approved, and this is John Hopwood with the first patient treated with MPS 6 enzyme. We flew him from Australia to Oakland, where this patient was getting treated, just for him to have the thrill of being there when the first patient was treated. And John, certainly opportunity to see this enzyme work he'd done for so long turn into a real product that's helping MPS 6 patients globally. One of my other great experiences was working in PKU, and I was able to work with Charlie Schreiber, who was and Dick Koch, were both BH4 people, but Charlie Schreiber also had worked on a PAL enzyme, basically ammonium lyase that could cleave phenylalanine. This was to treat PKU, where phenylalanine is elevated due to a metabolic block. And these were 2 strategies. One was a cofactor that stimulates the enzyme, maintains its structure so that it can metabolize the phenylalanine. The other is to give basically another enzyme that can help cleave phenylalanine if your own enzyme is deficient. Both strategies ultimately turned into products. With the BH4 program, we had to make a stable version is a thing that usually was stored at -70 in the freezer, but we're able to come up with 2 different companies to develop a room temperature formulation for it that would not degrade, and involved understanding science around the pterins and how to make them stable. And that was successful, and that's now known as Kuvan. The PAL, or phenylalanine ammonium lyase program from Charlie Schreiber, it was originally an oral enzyme therapy that he invented. But the truth is, oral didn't work. We had to turn it into a subcutaneous injected version, which was very potent. And that eventually got approved as Palynziq here. And that involved the company sort of recognizing the problem and switching gears. And I was on the company side trying to work with Dick and also Charlie on those 2 programs. But a thrill for both of them to see their science turn into something, but it took work to convert what we understood into something that could be used. Third example I'll show you is here, Peter Lobel at UMDNJ. He was a scientist, had been cloning various lysosomal enzymes. He discovered one, TPP1, and cloned it for Leiden-Patau-Batten's disease. That disease is a deficiency in this enzyme and results in a devastating brain disease. And we know the gene therapy attempts had failed. And part of the problem is you really need a lot of enzyme, and it's very hard to get that much enzyme from our current gene therapy strategies. We developed an intrathecal strategy, and so Peter wasn't didn't think that much of the intrathecal strategy, but it had one unique and important, valuable attribute, which was that it actually worked. And the intrathecal strategy I developed in my own lab while I was at BioMarin, and this was focused on MPS enzyme therapies, but it turned out it would work for CLN2. And so we took his enzyme and put it into the CSF fluid space and showed that we can treat kids with CLN2, and ultimately got approved as Brineura. Another example of a program. Finally, I'll talk about, the last one I want to talk about is a more recent approval. And this is for trihepnoid for long-chain fatty acid oxidation disorders. And this is Charlie Rowe. Charlie Rowe has been working in the area of energy, metabolism, and obviously highly relevant to the mitochondria. And he'd been studying fatty acid oxidation defects for years and had developed a treatment using odd-chain fatty acids, or C7 triglycerides, called trihepnoid. And C7 has a unique metabolism that will allow you to restore Krebs cycle function, which is, of course, a key part of the fuel cell that exists in your mitochondria. And he had studied informally for 12 years, treating kids with this compound, but had never gotten the sufficient investment to do a real trial and treat patients in a formal way that you could actually get approval for it. And it sort of stagnated for 12 years. Jerry Bockley was his colleague who also took over the program when Charlie retired. And we started working with Jerry then in 2012 and basically came on board to partner with them. They had the idea that what the drug is, how do we go from there to get to a place where the drug can get approved? And one of the things that Jerry told us was that, in fact, that patients didn't spend as much time in the hospital anymore. And we did some analyses early on of the patients treated during those 12 years that Charlie had treated, and were able to verify that there was a 69% reduction in the number of days in the hospital. We then conducted a trial, which we were able to verify that, and did another trial shown here on the right, where an additional 32 patients were treated. And you can see here how the number of days in the hospital, almost 8 days annualized per year, goes down to close to a fraction of 1, all right, and more than 90% reduction in the number of days in the hospital. And this is all with a compound that was kind of an oil-like thing, but it doesn't really matter what it is. It gets into your mitochondria, it gets metabolized and restores the Krebs cycle. And if you could do that for these kids, you can change their ability to produce energy with their mitochondria. That drug just got approved as Dojolvi in the US, in Canada, and in Brazil. And we're still working on European authorities to get it available there. And we'll be working on bringing it to Asia as well. That's one of the first mitochondrial treatments for fatty acid oxidation defects, which are fundamentally defects in mitochondrial function. So there's been great successes, and I think one of the things that's most exciting, new modes of treatment, not just enzymes or small molecules, but gene therapies and antisense oligonucleotides, maybe even mRNA therapies, so many different approaches. But one of the other challenges that's happening is more and more rare disease treatments are getting developed. There are also rising costs, and the rising costs are creating a problem in terms of how much has to be invested and how complicated it is. And there's lots of difficulties, expectations. I think these are going to be challenging for the mitochondrial disease area, particularly because they're a heterogeneous and rare disease. And when you're very heterogeneous, the system doesn't work for you very well. It just doesn't work. And I'll talk through some of those issues, both on study design and in use of biomarkers, which are really critical when you have heterogeneous clinical outcome diseases. And so we talk about CNS diseases, mitochondrial diseases, others of that kind. We need to get better at how we design studies so that the science that keeps coming forward actually is going to get approved and not get stuck in studies that are too small, not powerful enough, and are harmed by the tremendous heterogeneity, and which would cause us to think a drug doesn't work when it does. And this is, of course, the greatest sin in drug development. Now, to give you an idea of how much costs are rising and why this is a problem that the mitochondria community has to pay attention to, I thought it would be useful to show you what it takes to file an approval. And this is a part that's not seen to the academic world. They think, well, we've invented the drug and then you're just getting it approved. We have to understand that most of the time, the amount of money to develop an idea for a drug is a few percent of what it takes to actually get it approved. And the amount of money gets spent later is dramatic and crazy, really. And this is to put these applications together. I'll direct you just to look at the red box. That's the trihepnoid program, all right? We end up having a filing document for the FDA that had 228,000 pages, all right? 1,730 individual documents. There were 884,000 hyperlinks between one part of the document and the other part of the document. You have to imagine now how much work it is to put all these pieces together to treat a very rare disease. It is extraordinary, and it has so much complexity now, and it's getting worse, that in the end, it's going to make it harder to treat some of the rare diseases that are out there, even when we know how, and that to me is tragedy. But this hopefully gives you a sense for what it takes. So I want to talk a little bit now about what are those challenges in drug development, and challenges that will be very relevant to doing mitochondrial work. One is the study designs and the use of biomarkers. Study designs for rare diseases can have a very important impact, and the traditional randomized control studies can fail when you have high heterogeneity, and especially with a lot of irreversible disease. If people can't get better and you're trying to prevent decline, then there is a real problem when you have heterogeneity in terms of how much disease is present and how much of it's irreversible. And we think alternative designs can be far more powerful and more valuable, and I'll show you some examples. If you can use biomarkers, it greatly helps because clinical endpoints involve a series of things. There's something wrong biochemically, and then it has a second effect and a third effect. And when you measure things clinically, you might say, well, that's what's important to patients. Well, it is. The problem is that it's noisy, it's not precise. And therefore, if you're talking about 20, 30 patients in a trial, it's very hard to use noisy endpoints. And then you can't make good decisions. Is the drug working? Is it not working? So biomarkers, we need to get better and smarter. And they're not just a compromise. They are actually going to be more accurate measures of disease activity and are important in actually succeeding to develop potent drugs. Now, if you look at study designs that are out there, there are certainly many different types of studies. I look a whole group of them here: randomized studies, natural history controlled, crossover designs. I'll touch on a few of the others. Everyone believes and knows that randomized double-blind placebo-controlled studies are the gold standard. But while they're the best designed, they're not always just rigorous. Sometimes they're just insensitive. And we can't tell the difference between what's rigorous and what's just insensitive. And I think this is where we have to get smart about recognizing when that is, particularly when there's heterogeneity at baseline and a variety of different genotypes or disease state consideration for a disease. So let me talk about some of these other designs. Now, many people think, oh, well, we'll just do a natural history study and we'll use that as control and we'll get FDA not to make us do a placebo-controlled trial. The truth is, it's very hard to get approval with a natural history, an independent natural history control arm. This is one example I just talked about before, Brineura, an enzyme therapy for CLN2. But keep in mind that the natural history control shown there in red is very tight, consistent. These patients go from no symptoms to being devastated, 90% loss of function in basically 2 years. It's a very rapid decline. In that setting, with rapid decline, you can do this kind of a study. But for most of the other disorders, it's not as fast as that, and it's not as consistent as this. And therefore, studies would have to go 5 or 10 years, all right? So that just is not going to be plausible. And so this type of design people want to do, but I'm just be honest with you, it's very hard to get regulators to accept this, except if you have very rapid decline where they can really show the difference in a short period. Now, what I've tried to promote more is the idea of crossover designs. That is, a crossover design is where you take a group of people, very heterogeneous, monitor what they're like, right, over time, cross them over onto treatment and compare their before and after. FDA doesn't like this mainly because other things could have changed when you cross them over, and I get that. And so the problem, though, is that when you do it a crossover design, you help remove variability factors because that patient's being compared to themselves. Wherever they are in their disease, you're looking at how they did. Now, you need to have good ascertainment of what's wrong with them in the run-in period or in the retrospective run-in period. So you need to have high quality ascertainment. And you have to make sure when you're crossing them over, you didn't change things, certainly. And whatever you're measuring needs to be objective because it's not blinded, so you have to have an objective measure. And generally, if you're going to make crossover studies work, the effect size should be large. It still should be a big effect on an individual case basis. I put below an example. It's not a mitochondrial disease, but it's a bone disease, X-linked hypophosphatemia. We did a phase 2 study and we used blinded x-rays of before and after to show the improvements. And ultimately, when we did the phase 3 trial, it verified what we saw in an open label crossover design. And by blinding the readings, we could actually allow ourselves some objectivity in this particular design. Now, Tribenoid was another study we did a natural history control. In this case, the trial period was an 18-month retrospective period. And remember, we have in this study were 5 genotypes with all different types of severity and ages. Young kids, adults, all mixed up in one study with all these different genotypes. This is a little bit more like what your situation might be in a mitochondrial disease. We could enroll 29 patients. We look for patients that were having a lot of hospital events. They're in the hospital having trouble. And the idea is, does that change if you cross them over on Tribenoid? What we show here, these are actually the 29 patients' lines, and you can see what happened to them afterward. And we actually had a 50% reduction in number of events, the number of days in the hospital, but more importantly, if you look at it from a median standpoint, a 77% reduction in the median number of days in the hospital for the 18-month period before you crossed over to after. And it didn't matter what type you were, whoever got enrolled, we're doing the same comparison. So the power is dramatically better because there's no randomization problem. You don't have to match the groups. They are matched to themselves. Now, in that trial we had based on the retrospective study of those patients Charlie Rowe had treated over 12 years. We then took the hypothesis that there was this change in major clinical events and major clinical event days, ran that study and showed in 29 patients that we actually had statistically significant reduction in the events. Now, if you continue to follow those people in extension, those events got even better in the second 18 months, giving you confidence that this is real. This is not a temporary excursion. They kept getting even better in the second 18 months, all right? So 3 years of treatment time. Now, if you look, we had enrolled another 32 patients actually in an extension study who were also needing compassionate use. We enrolled in the study and actually got turned out to be more than 90% reduction in their event rate. And more recently, Dr. Gouffon, Dr. Corral did these for French and Austrian patients, respectively, also showed the same dramatic reductions by crossing over before and after. So it's really 5 different populations of patients now have been shown this crossover effect. Now, despite that, we had a great difficulty getting regulators to accept this because it wasn't randomized control. But to do a randomized control study would have taken dramatically more patients. In fact, because we had so many different genotypes and so much variability in the manifestations, it would have taken us potentially more than 120 patients, which would have dramatically altered the cost of the program. So we were able to get approved in the US, and we're still working our way through getting it in Europe. Now, the last design I just want to touch on is a novel design we call the blind start design. I think it's an interesting one when you have a really small population and you want to do something placebo controlled, but you can't run a traditional randomized study. In this design, you randomize to 4 groups. The first group gets on active treatment right away. The other groups cross over on active treatment at intervals of time. The patients don't know when they're crossing, so it's blind start, meaning they're blind to when they start treatment. They know they're going to be on treatment at some point, but they don't know when. And you actually compare each baseline for each group, which is the last assessment before they cross onto drug, compared to 24 weeks of treatment of drug. This is the design we used in the MEPSEVI trial for MPS 7, it was an enzyme therapy. The thing about this design is that when we model it running trials, we can show that this is 5-fold more powerful than the same size study as a randomized control study, 5-fold more powerful. And that is hard to understand. Why is it so much more powerful? Well, it turns out it's more powerful because of 2 things. One, all patients get treated, so you get treatment effect size estimate in all patients. But the second thing, even more important, is that you never have an abnormal randomization or a skewing where all the sick people are in one group and all the mild are in the other group, or all the old are in one group and all the young in the other group, or other kinds of stratification, which is very hard in a little study. If you have 12 or 24 patients, trying to control randomization so it's balanced is extremely hard, maybe impossible if you have a lot of genotypes. And so each time you run the study, since everyone gets treated, it always works. You're always doing the right comparison. That gives it 5-fold more power. That is not trivial. That is a big deal when you're talking about rare disease drug development. We think this is a good design when you can't do a randomized control study. The regulators accepted it in Europe and the US for MEPSEVI, but it's not exactly an easy acceptance. But I think it's a type of design when you're doing an ultra small disease where you can get the most information out of a group of patients. Now I want to talk about biomarkers. I don't want to spend too much time because our time will be running out, but biomarkers are a really important part of the story too. And when you have significant clinical variability, you have to be able to look at what I would call primary disease biomarker, something that measures disease activity at the beginning of the disease and be able to show if your treatment is specific for that disease, have an impact on that marker. That will tell us more about what you're fixing in the biochemistry of this disease. The actual clinical outcome will depend on where that patient is in their course of development. Are they early? Are they late? Are they severe? Are they mild? The challenge right now is that many patients have severe or substantial reversible disease, and that's true in mitochondrial disorders. And what is terrible, the catch-22, is that you can't get approval. If you can't get approval in the biomarker, then you're waiting for clinical endpoints. But if you already have a lot of irreversible disease, well, you need to screen before they become evident. But you can't do that because they won't do newborn screening unless you have a treatment approved. Since you can't get treatment approved, you can't do newborn screening, you'll never find the patients who are not already past points of irreversibility. This is a challenge that we need to get over and grow up as a society and start understanding how to approve drugs on biomarker-based approvals, catch them by newborn screening earlier, and then devise and evaluate carefully what's going on over time. We need to change how we do it, and this will change the future for rare disease, including mitochondrial disorders. Historically, it's been very hard to get biomarkers approved. We did this paper a few years ago just to show within a period of 5 years sampling that all 11 approvals, which FDA said they did do biomarker-based approvals for rare genetic diseases, well, all of them were based on biomarkers already in existence from the 1990s. There was not a single new biomarker in that 5-year interval. And since 2014, there have been a couple biomarker-based approvals, but it's extraordinarily hard. And for some, like metabolic diseases like Sanfilippo or Niemann-Pick type C, there should have been acceptance of the biomarkers, but there hasn't been. I think this is a challenge for us going forward. If you could accept a biomarker-based approval, here's what the benefit to be to society. If you had $1 billion to invest and you're using a one study surrogate endpoint or biomarker endpoint, you could actually develop 36 drugs. And this is based on an evaluation we did of 15 different diseases where there was an animal model data of the efficacy, and we designed clinical programs with or without a biomarker endpoint. If you use the traditional clinical endpoint approach, you could only develop 11. So it's almost fourfold more drugs, right? If you were able to use biomarkers, think about how that changes things for all of us if we could develop way more drugs. And even if some of them don't prove to be clinically important because the biomarker is not accurate, and we lose some of those, the overall effect will be a dramatic improvement in innovation and treatment, including for mitochondrial disorders. For MPS 7 enzyme therapy, where there was less, like 100 in the world, 200 in the world at most, we need to use biomarker approval. EMA accepted. This is the biomarker of urinary substrate. I call that a disease activity biomarker because it's the thing that builds up in front of the block, the enzyme deficiency block. And we could show it being reduced in the urine and blood, and we could show in animals how that correlated with the disease state. But if you look at the walking in this trial of 12 patients, it turned out that only 3 of the 12 patients improved in their walking. And everyone said, see, the biomarker doesn't prove anything. Well, first off, 6 of the 12 patients had degenerated hips at the start of the program. So if your hips are completely degenerated, what is the possibility that enzyme therapy is going to fix them? It's 0. So just like irreversible neurological disease, those 6 patients will never read out walking. If you look at the actually only 6 patients that did walk, 3 out of 6 responded, which is actually the same, and they had 3 very good responses. Those were very much the same response rate as for other randomized controlled trials that have been done in MPS diseases. So the data really are the same. And if you use the biomarker, you get the right answer. But clinical endpoints are not just dependent on whether you can improve walking, but whether you are improvable at all by any drug. And that heterogeneity in disease state is what's killing us in neurologic diseases. And this is just a way of looking at it in MPS 7. I put it in this diagram here. It's maybe a little complicated, but on the y-axis of the graph above is the reversible disease. When you start, let's say you're the blue line, you're a severe patient, you first you don't have that much disease, your disease accumulates, and then it reaches a point where you start getting irreversible disease and the amount of reversible disease goes down. So think this is the curve of reversible disease. A severe patient's blue, the brown is intermediate, and red might be a slowly progressive. If you do the biomarkers at 3 points in time at these ages, the biomarker will show you the same effect, that you're reducing it. What happens clinically to those people, though, will be dependent on how much disease they have and how reversible is it. And this is just to give you schematically why the biomarker gives you the right answer, but depending on what age and which type of patient you're touching on a particular endpoint, you'll get all kinds of different clinical results, right? This is try to help you understand conceptually what the problem is. So we need more accelerated approvals for rare neuromuscular disease, particularly for mitochondrial diseases, and we need to change how we're approaching the system. So what we try to focus on is what I call primary disease activity biomarkers. These are the ones at the beginning of the process that are most closest to the defect. I think those markers are the ones that are much more predictable, have predictable value. Things that are downstream due to pathophysiologic processes are much more variable, less reliable. But I do think we can get better about finding more primary disease activity biomarkers, and being able to develop drugs that way will change the future for many neuromuscular diseases, including mitochondrial. When you look at neuromuscular diseases, and this is that part of the irreversibility story, if you're early in the course, you can't show improvement because there's not much improvement to had. And you're really trying to show that if you didn't, against a natural history control, there would be a difference. And that's only if it happens quickly can you do that. And here, if you, as time goes on, as health goes down, they get diagnosed, and let's say they're 7 or 8 years old, the ability to reverse can be dramatically altered. And so then what you're looking for is a small improvement, but a variable improvement depending on where they are in this decline curve. So the neuromuscular challenge without a biomarker is incredibly difficult, and you'll need a large number of patients, and very few people can afford to do that. And even when they're large, it's very hard to overcome the kind of heterogeneity that exists. So we are not doing as well as we could. So I'd like to end then by talking about rare disease treatment development. The academic work has been so important at opening the door to ideas for treatment And from there, the industry can pick that up and turn that into an actual approved drug through incredible amount of work, incredible amount of documentation, paperwork, and other things, and can struggle through the regulatory process. If we can improve the efficiency in how we do this, we can get many more treatments developed, and particularly for the disease now, we're not getting enough treatments developed, like mitochondrial disorders, where they're very heterogeneous, they're variable degrees of irreversibility, and the system doesn't work for them, and we need to change that system. Alternative study designs are one way. Crossover type designs, blind start type designs, I think are one way that we can move forward. We need to get the regulatory authorities and all of you, academic world, to understand and support that. And we need to try to modernize our views of proximal disease biomarkers, the ones at the beginning, and the valuable and the high predictive value they have for rare disease drug development. And if we can start improving treatments with these very profound disease activity biomarkers, I think we can change the future for what happens in rare disease, including mitochondrial. And if we do this, we need to do high quality long-term follow-up for sure to make sure that those treatments are the ones we want. If we can make these improvements, I think we could translate more of those great academic discoveries into actual treatments for our patients with all kinds of rare diseases, but most importantly also the mitochondrial disorders. So I want to thank all the patients, investigators, and others who helped us with the work over the years, all these different academic investigators who I had an opportunity to work with and make their dreams come true. And nothing more thrilling to be able to be on one side of that equation and say, hey, we're going to come and invest money in your idea and turn it into something that people around the world can get. And there's no greater finish to a career to be able to know that you've taken your science, 20, 30 years of it, and turned it into something that people can use. So thank you for your time today. Appreciate your support.